Hi, my name is Arale Castillo, and our focus today is the cell cycle. The cell cycle is basically a series of changes that a cell undergoes from the time it forms until it divides. Cell division occurs from a series of precisely timed and carefully regulated stages of growth, DNA replication, and division that produces two identical daughter cells. And as seen in the diagram, we have two major phases, which are interphase, in which the cell is growing, DNA is replicated, mitosis, which the replicated DNA and cytoplasmic context separate and the cell divides. Different cells take different lengths of time to complete the cell cycle. A typical human cell might take about 24 hours to divide, but fast-cycling mammalian cells, like ones that line the intestine, can complete a cycle every 9 to 10 hours when grown in culture. So we'll dive into interphase. It's a very active period as the cell is undergoing normal growth. Internal and external conditions must be met to enter the mitotic phase. And we have these three stages that are based on the sequence of activities that are occurring. And the first one is the G1 phase, or the first gap. Cell is physically growing larger, copies organelles, and makes molecular building blocks needed for later. And then S phase, or synthesis of DNA. The cell is making a complete copy of the DNA in its nucleus. The centrosome, a microtubule organizing structure, duplicates, which helps separate DNA during the mitotic phase. And then lastly, the D2 phase, or the second gap. Cell is growing more and more. It makes proteins and organelles and organizes its contents in preparation for mitosis. Mitosis constitutes a form of cell division that occurs in non-sex cells or somatic cells and produces two identical daughter cells. It's basically this multi-step process in which the duplicated chromosomes are aligned, separated, and move into two identical daughter cells. In the case of humans, we have 46 chromosomes, which are two sets of 23 chromosomes with one set inherited from each parent, as opposed to meiosis, which is another cell division that occurs in cells that give rise to sex cells, and we have such as sperm and oocytes, and we have meiosis halves the number of chromosomes to 23, and when the sperm fertilizes the egg, the total number of 46 chromosomes is restored. And we have the first portion of the mitotic phase is karyokinesis, which the nuclear contexts divide, and the second portion is cytokinesis, which is the physical separation of the cytoplasmic contents into the two daughter cells. So we'll focus on the major stages of karyokinesis or mitosis, which are prophase, prometaphase, metaphase, anaphase, and telophase. The first one is in prophase. Chromatin condenses into chromosomes. Centrioles move to the opposite sides of cytoplasm, and the nuclear envelope breaks down and nucleolysis disappears. The prometaphase, chromosomes continue to condense. Proteins attach to the centromeres, creating the kinetic cores that appear at the centromeres, and the meiotic spindle microtubules attach to the kinetic cores. Centrosomes move toward opposite poles. Metaphase, spindle fibers from the centrioles attach to the centromeres of the sister chromatids of each chromosome. Chromosomes align midway between the centrioles. Anaphase, centromeres separate and sister chromatids move apart, with each chromatid now an individual chromosome, and spindle fibers shorten and pull these new individual chromosomes toward the centrioles. Telophase, chromosomes arrive at opposite poles and begin to condense. Nuclear envelope material surrounds each set of chromosomes, and the meiotic spindle is breaking down. Finally, cytokinesis, which will lead to these two identical daughter cells. It's actually beginning during anaphase, when the cell membrane starts to constrict around the middle of the cell. But we have this constriction called this cleavage fur, which continues to tighten throughout telophase. The contraction of a ring of microfilaments pinches off the two cells from one, and then as the ring is pinching off, it separates two newly formed nuclei and divides about half of the organelles to the daughter cells. And these newly formed cells may differ slightly in the number of organelles in size, but they have identical chromosomes and contain identical info. This is my works cited, and thank you for watching.